So welcome to CS Medicus, independent information about science and practice. My name is Dr. Dirk Seeling, and I'm delighted to be speaking with Professor Gerald H. Pollack from the United States of America. In our 30 to 45-minute conversation, we will focus on the fascinating potential of easy water, especially regarding cellular detoxification, energy, and cell metabolism. We will also try to think about possible effects for patients with post-COVID and post-vaccine syndrome. Gerald H. Pollack is a professor of bioengineering at the University of Washington in Seattle, where he has been a faculty member since 1968. He is renowned for his groundbreaking research on water in biological systems, particularly the discovery of the exclusion zone EZ, a fourth phase of water that plays a key role in cellular function and energy dynamics. Professor Pollack is the founding editor-in-chief of the journal Water and serves as executive director of the Institute for Venture Science. His work has earned him numerous honors, including the Prigogine Medal, a National Institute of Health Transformative R01 Award, and honorary positions with the Russian Academy of Science. He is the author of influential books such as Cells, Gels, and the Engines of Life and the Fourth Phase of Water, which bridge biophysics, cell biology, and innovative water science. I'm very delighted to welcome you, Professor Gerald H. Pollack.
Well, thank you, Dirk. I'm delighted, thrilled to be with you.
Thank you. So what is easy water and how does it support the body's self-regulation and health?
My goodness, you ask a very broad question. Maybe it's best if I start historically, because that may provide an accurate picture of what you're asking. We were initially involved in a different field, the field of the molecular mechanism of muscle contraction. And we hardly thought about water, even though, water is by far the main component of muscles. When people think about muscle contraction, they think about the proteins myosin and actin, but it's as though those proteins act in a vacuum. There's no water. I have rarely seen any model of muscle contraction that takes into account the fact that it's mostly water. It's 2/3 water, like roughly most tissues. And, if you translate that volumetric ratio into the numbers of molecules. In other words, you take all the molecules inside the muscle or any cell, and you line them up one by one, and you start counting. It turns out that more than 99 out of 100 are water molecules, and the reason for that is, the 2/3 volume. If it's filled with water, the water molecules are so small that you need a whole lot of them to fill that 2/3 volume. So when you think about it, you think about that the muscle is mostly water, 99 out of 100 molecules, and nobody takes into account the possibility that water may play a role in muscle contraction. It's completely ignored. It seems preposterous in retrospect. And I guess I must admit that I fell into that category. But one day at a meeting, a conference that was held in Hungary, to which I was invited, and the conference was designed to commemorate the work of a famous Hungarian biophysicist who had passed. His name was Ernst, and he had two interests. One was muscle contraction, how it worked at the molecular level, and water. And of course, I had no particular interest in water, but I presented our work on muscle contraction. And I think it was, reasonably received. And then some guy came and presented on water. His name was Gilbert Ling, and he's known among some people, but lesser among others. He passed 5 years ago, and Ling came from China. He was chosen in 1948. The Chinese government decided to send the most promising young scholars, Chinese scholars, to the U.S. to study. There were four of them. Ling was one of the four. Three of them went on to win Nobel Prizes. And Ling, in the same category of people many think should have won at least two Nobel Prizes, but he was passed because his ideas were so radical. So what were his ideas? Well, he said that that water inside the body, or inside the cell in particular, is not like water in a glass. You read a biochemistry book or a cell biology book, and chapter one always talks about water. And the idea is that the water in the cell is liquid water, liquid. Um, and Gilbert Ling said, no way, it's not correct. And he said that it was more, although he didn't use this particular word, but I think what he was alluding to was it's like a liquid crystal. In other words, the molecules were all lined up like soldiers at attention.
Mhm.
Now, this is a radical view because if it's true, since all of biochemistry and cell biology is rests on the premise that the water inside the cell is just ordinary liquid water, everything is different. And he produced a lot of evidence that it really was the case. And I came away mesmerized. I was really blown away by not only by his idea, uh, but also by his evidence, and evidence that other people had produced and presented at that conference. Well, I didn't trust myself. I came back to Seattle and I gave one of Ling's books to a few of my students, the more astute ones. And I said, do me a favor, read it and tell me what you think. So they read it, they came back to me, and their response was uniform. They said, this is so interesting. Uh, and if correct, it means all of cell biology and biochemistry is wrong from the foundation. And he's presented a whole lot of evidence that he's right. So first thing I did was to write a book, and that book it was Cells, Gels, and the Engines of Life. The purpose of the book was twofold. It was mainly to present Gilbert Ling's ideas that the water inside the cell is different, to present it in a way that mere mortals like myself could understand it, because Gilbert Ling, one deficiency he had among a bunch was that he was brilliant, and he presumed that everybody else was equally brilliant, and and that no matter what he wrote, everybody would be able to digest it in the same way that he can digest it. I think I think I think that he never really understood the importance of editing. And I kind of think maybe in the Chinese language, maybe the word editing is omitted. I'm not sure, but his writing was difficult. So I tried to remedy that by producing the first half of the book was designed to present Gilbert Ling's ideas in a kind of simplified form that could be digested by ordinary mortals like myself. I'm not sure if I succeeded or didn't succeed, but Gilbert Ling didn't like it. I think he felt that I stole his thunder, and that um, uh, but whatever, it's not important. It took me 10 years to restore our our friendship when we came up with evidence for long range ordering of the water. he liked that. Anyway, and the second half of the book was designed to answer the question, well, so what if the water is ordered? What does that mean for function? And I was able to deduce from evidence in the literature that the cell undergoes a phase transition. That is, phase transition, biologists are not so familiar, but chemists and physicists know about phase transitions. It's from one state to another state, like gas to liquid or liquid to solid.
Yeah. I think it's for our participants. So if we have water, it can go to ice and it can go to air. Yeah, so
gas.
everyone knows about that by in the kitchen or when it's cold.
Yeah, that's the central idea. And what I found out and presented in the second half of the book was that in at least a half dozen different cell types, I could demonstrate the evidence that that happens in a cell. In other words, the cell goes from a structured, that's what Gilbert Ling called it, structured, ordered, structured water, undergoes a phase transition from the structured water to ordinary liquid water, and then back again to the initial state. And as the water undergoes that transition, so do the proteins inside the cell that are largely responsible for function. So for example, in the case of muscle cells, since I know a little bit about that, when the muscle is relaxed, when it's not contracting, the water is structured. And later we found out that the structuring was different from what Gilbert Ling was talking about. But I'm just giving you some background. It went from structured, and when it's structured, when it's ordered, the proteins are kind of confined and in their extended state. They can't do anything. But when the transition occurs and the water changes from this highly structured state to ordinary liquid water, the proteins undergo a folding from this extended state to the folded state, and then they go back again. It's that transition of the proteins that's responsible for the cell function. So, for example, in a muscle cell, when the muscle's relaxed, the proteins are in the extended state. And then when the transition occurs, when the water transitions from the structure to ordinary liquid water, the proteins fold. And it's that folding that's responsible for the mechanical contraction. And then back again. So that.
So may I ask one question? So
5 questions.
yeah, what I understand, it's not much what I know, but it's also it has to do with biophysics, electronics, that contraction is happening, and how does it? What is the concrete effect it has on water and on the concrete cell when there's electrical charge?
Well, the electrical charge turns out to be central, and that's what was missing in Gilbert Ling's thesis, the electricity. So we could confirm experimentally that there was order. Since we have limited time, I won't go into detail, but what we found out that this structured or ordered water, which we call fourth phase water or EZ water, exclusion zone water, was charged. It's not neutral, you see. And when it formed, we could observe it in the laboratory. When it formed, we were curious to see if it had any charge. We didn't expect any charge because, you start with ordinary water, which is uncharged. And then we started with water in a chamber, and then we could observe this structured kind of water, and we would expect that it would be neutral as well, but it wasn't. We stuck an electrode in, and we found that it was typically negatively charged. Now that doesn't make any sense at all, because if you start with no charge, how do you wind up with a large region structured water that has charge.
Mhm.
So the only conceivable idea is there must be another region that has an opposite charge, equal positive charge, and that's exactly what we found. So we found that the EZ or fourth phase water was negatively charged, and the region immediately next to it was oppositely charged.
Mhm.
And so when you think about that, a region of negative charge next to a region of positive charge, you have a battery.
Yes.
And we could determine in vitro, that is in the laboratory, that if we put one electrode in the negative region and one electrode in the positive region, and we connected them to an LED lamp, we could light the lamp.
Oh, really?
Yeah. In other words, it's an energy source.
Yeah, great. Mm-hmm.
On the other, now, another critical question that we were able to answer is, uh, what supplies the energy to this battery? All batteries need to be charged. Your cell phone, if you don't plug it in at night, you wake up in the morning and you have no useful cell phone,
Yeah.
right? And a car battery is getting charged all the time. So um, you need to charge all batteries. And the question is, this battery that exists, the negative next to positive, how does it get charged? Well, it turns out that a student in the laboratory got the answer. We couldn't figure it out. How would we know? We're naive scientists, and this is new territory. Well, so he was experimenting with an experimental chamber in which he could visualize the negative and positive charges, the structured water region and the region beyond. And he found a lamp right next to him. And he shined the lamp on the chamber, and the region that was illuminated, the region of negative charge, the region of structured or EZ water, expanded hugely. And then when he took away the lamp, it came back to the original.
It
So
was responding to light. Yeah. Mm-hmm.
light,
Yes.
exactly, light. It was light. And of course, we did follow-up studies, and we found that some of the wavelengths had no effect. It was an incandescent lamp, so it had many different wavelengths, and we wondered which ones were the most effective. It turns out that red light was moderately effective, and infrared light was supremely effective. So the longer wavelengths were the ones that could build this battery's charge, was responsible for charging the battery. And when you think about it, there's precedent in photosynthesis.
Yeah.
Where does the energy come from in photosynthesis? Green plants, what is light?
Yeah. Mm-hmm.
And the first step in the, I think, 20-step sequence of steps in the photosynthetic process, right at the beginning, first step is light comes in and separates charge in water. And that's exactly what we found.
So this the wide wavelength is giving structure into water, right?
Yes, that's right.
And short, if it is like zzzt, short wavelength is destroying water or
No, it
structure.
doesn't destroy. We found very recently, we found that it actually builds charge in the battery. It is negatively charged exclusion zone, easy structured fourth phase water becomes more charged when exposed to ultraviolet light. But it doesn't increase the size of the zone, but it increases the charge of the zone. So you might call it maybe a different mode of energization that occurs. But light is critical. And, it's not a new story, because we know in the plant kingdom and also some single cell organisms, light is critical. So you might say there's nothing new. But I think it is new in that we understand the process of how it occurs, and we think that photosynthesis is maybe one prime example of the more general phenomenon that we discovered. It's probably nature's ideal or most effective or most efficient example of this process that
Mhm.
we discovered. So
Of
it's nothing
life energy, yeah. It's
life
really.
energy. Yeah, yeah.
Mhm.
And we found evidence that you, Dirk, and I use this energy in our function. Um, if you, I don't know what other questions you have, but I could tell you about the evidence for this if you want to.
Yeah, it would be very interesting to go to this. Why is the structure of water? And as we heard, it's a hexagonal phase. Yeah, it's a
Oh,
hexagon.
yeah, it's a hexagonal, but let me tell you about specifically about the energy
Hmm.
uh, example of your use of this kind of energy that we're talking about
Yes.
for
Hmm.
body function.
Yes.
It started in our laboratory where we took a a tube, a cylindrical like a straw made of a material called naphion, uh, and this naphion builds very nice exclusion zones right next to it. So we use it pretty commonly. So we took a tube, like a straw, and horizontally we immersed it in water. And what we found is that there's flow that goes through the tube and it never stops. It just keeps going.
Well,
So we were astonished. This was another discovery by a student, a young student in the lab. Some of these young students are very clever, and they're more
yeah,
clever
because
than
they have no blocking of theories. Yeah.
right. You're exactly right. Yeah. So, uh, uh, I'm constantly impressed by the creativity of these students. Anyway, um, so we found eventually that the reason for this is if you think of the cross section, what happens is that's made of naphion is right next to the naphion, just inside an annular kind of a structure, the EZ forms. And now it's negatively charged, and next to it is positive charge. So it's the core of the tube that these positive protons are lying.
Yeah.
Uh, Now these are free to move. The negative EZ is stuck to the surface, but the positives are free to move, and so um, they ordinarily latch onto water molecules. This is well-known chemistry to give you a so-called hydronium ion. So it's kind of like positively charged water. So the core is filled with this positively charged water. The molecules repel each other. They want desperately to get out of the tube because of repulsion, and they exit either that way or this way. So if they exit that way, water is moving out and it gets replaced with new water, and it's perpetuated that way. So that was the original finding, and we didn't at the time we didn't know really about applications of this. So it was, I think, the next year I was invited to Russia, and I have a lot of friends in Russia. I really respect the Russian scientists, and I was visiting my friend Vladimir Vyakov, who runs the biochemistry department in Moscow University. And we were sitting and enjoying some caviar and vodka and whatever. And then he says, "Well, maybe let's go to the laboratory." So we go to the laboratory, and he said, The first thing is I want to introduce you to my colleague down the hall." So he calls his colleague. His colleague comes in, and in Russian, he speaks no English, but Vladimir is a great translator. And he says to me, "There's a big problem in the cardiovascular system." I said, "Well, what kind of problem?" I was maybe feeling a bit arrogant because I did my PhD thesis on the pressures and flows in the cardiovascular system, and
Oh,
I thought we really understood pretty much the basics. But he said, no, 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 there's a big problem. So in great skepticism, I asked, well, what's the problem? Uh, and he had me convinced in 5 minutes that he was right and I was wrong. Uh, and so what's the problem? Well, it's a really obvious problem that people don't think about. Um, the red blood cell is 6 or 7 micrometers in diameter. The capillaries go down to 3 or 4 micrometers in diameter. They're pretty small. To get a red blood cell to flow through a capillary, you have to squeeze it. Um, and you can see video evidence. We have it. Other people have it. That when the red cells are going through the capillaries, they bend, they squeeze, just like this is what
yeah.
they look like a backwards C,
Mm-hmm.
like that. Now he said, well, this requires energy. It's sort of like sometimes your toilet gets stopped and you have to take a plunger and you have to push to get it through. It's the same principle. It takes energy to push it through. So he calculated, he said, if if the left ventricle is responsible, the pressure developed by the heart is responsible for has enough energy to compress all of these red blood cells. He calculated that the amount of pressure that would have to be developed is not 120 millimeters of mercury or even 180 or 250 or any, but it's 1,000,000 times that. Now, so he said it's impossible. He said unless the heart can really develop a quote high blood pressure, there's got to be something else that complements the heart.
Mm-hmm.
Uh, and he starts going off on a whole lot of different ideas. And I must admit, I wasn't listening intently because I just was thinking that in the laboratory, we just found that if you have a tube, that flow goes through the tube spontaneously. And the energy for that is infrared energy because that's what drives the flow in the tube. I was talking about building easy water and such. So I went home and I asked one of my students to follow up on this, to ask whether that extra feature in the cardiovascular system might be what we discovered in the laboratory. So he went ahead and he told me afterward, by the way, after he had a successful result, he said, when I came to him and suggested that problem, he thought I was on some kind of drug. uh, anyway, so the key feature of what we found in the laboratory was that the energy comes from infrared energy. So he did an experiment on the cardiovascular system of the chick embryo. And a 3-day-old chick embryo has a pretty well developed cardiovascular system, but the regulatory systems are not yet developed. So it's the right kind of preparation. And he managed to be able to follow the flow in several different blood vessels. So step one is he stops the heart. And so the heart's not pumping anymore. It's just a blob, in the way of the flow, by the way. And still he found that flow continued, but it didn't. It was a modest flow, but it continued. It didn't stop. And everybody would expect you stop the heart, the flow stops, but it doesn't stop. And my student found no fewer than a half dozen publications scattered throughout the previous century that showed exactly the same, that you stop the heart and the flow continues,
Okay.
which is very interesting and completely unknown to to most people, even most people studying the cardiovascular system, but it's there in the literature. And he was able to confirm that. And then his main experiment was, okay, let's take the flow that's existing and add infrared energy, because that's what drives what we found in the laboratory. And he found that the flow increased by 3 times or more.
Wow. So the charge is coming from light and also for the blood cells for
Yep.
this flow in the cardiovascular system. Yeah.
It appears that way. So it means, well, it means a couple of things. It implies that in your cardiovascular system, and maybe even mine, um, that it's not just the heart that pumps the blood, it's also the blood vessels
Yeah.
that pump the blood. Just like the tube in the laboratory, the blood
Yes.
vessels are pumping blood. And
Yes.
if your heart stops, it doesn't mean that your flow stops. And maybe that implies you're not dead.
I mean, Gerald, the one of the problems of the what they said about mRNA vaccines was that the charge of the vaccines was not as they told us. So because of that, there was a magnetism from the vaccines to the vessels, and so it was stucking in the vessels. And this is so it is of course a charge problem also. What is in the vaccines? If it is flowing, if it is helping the blood, or if it is stucking at the vessels.
Well, I'm
And
no
Mhm.
expert in this, so
Yeah.
I don't have, but I have heard of some clots that appeared.
Yeah, yes, that's it. Because that's why they stuck there, and then then they catch up what is flowing through the endothel cells, and
Yeah.
then they're the plugs. Yeah.
Well, that's very interesting. You know, uh, if we're right about easy water and the importance of easy water and the
Mhm.
universality of easy water, it's
Mhm.
going to have implications for just about everything.
Mhm.
Not just the vaccines, about which
Yeah.
I don't know much, but it's literally everything is
Yes.
going to be impacted by it, and 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 that all of that remains to be elucidated. Um, there's also, you might say, a sort of a general phenomenon. If I may expound a little bit on this, because um, the general phenomenon is about function. So I mentioned that cell function depends on a phase transition. So let me amplify a little bit. So you have a cell, and the cell is, first of all, filled with easy water.
Mhm.
And I said it has negative charge. And if the cell is filled with negatively charged easy water, of course the cell is negatively charged. Now, the usual interpretation of the reason why the cell is negatively charged has to do with membrane pumps and channels. If you pump more negative charge into the cell than you pump out of the cell, then you get a net negative charge. I think this is absolutely wrong. And there are a half dozen different reasons why that idea is erroneous. And I've published on this, anybody who's interested. A simpler explanation is that the cell is just filled with easy water, and that's why it has a net negative charge. And most
Mhm.
cells are between minus 50 and minus 100 millivolts negative. They're always negative. And when the cell undergoes so-called action potential, when it's stimulated to do something, this negativity disappears, and it goes, the potential difference goes from negative to zero, and that's called the action potential. But it's according to the thesis that I'm putting forth, it's a direct result of this phase transition. Um, so
Mhm.
uh, so now, if a cell is filled with easy water, then it has a large, robust magnitude of electrical potential, let's say minus 70, minus 80 millivolts. But it turns out it turns out that cells that are dysfunctional or even pathological, instead of minus, let's say minus 80, they're minus 40 or 30 or 50. And why is that? So according to the thesis I'm putting forth, it's because the cell doesn't have as much easy water. It doesn't have as much energy because the energy is stored in the water, in the easy
Okay.
water. And if it has a even smaller magnitude electrical potential, minus 20, minus 30, then it's cancerous.
Okay, okay.
And then
So
if you
we
put
have to maybe just to tell, because we didn't say what is exclusion zone. So it means that particles are extracted out of this exclusion zone. Yeah, you tried this and with different toxic things, and always this water is excluding it. That's right.
is virtually everything, right? Because it's a liquid crystal, and in order to gain the purity that it shows, it has to get rid of everything that had been included in it. So this is certainly one way to get rid of contaminants, toxins, whatever.
And this was replicated in different laboratories all over the world, or is it only in your laboratory?
Oh, no, there are laboratories all over the world that replicated uh, some aspects of what we found. I can think of at least two laboratories, but I think there were more. We've done so much in the last 20 years that I don't say that any laboratory replicated everything we've done, but the essence, yes, has been replicated. Yeah.
I'm just because I read about you, and there were the typical web pages. I know already all innovative scientists who are not with the mainstream scientists. They are collected in one web page where you can read about them, that they are crazy, like that you got under drugs, or that you got a coconut on your head or something like that. And I'm always saying, okay, I try my best to come into this web page, but still I'm not there. But it is, it's great that you because you have different laboratories, and you can, of course, the evidence is there if there are more than one laboratory showing that there is an easy zone and there is a special phase of water. So what are we discussing about? Yeah.
Well, yeah, it's difficult for me to respond to that. We all know that, there are sociological or psychological issues that, um, uh, yeah, every scientist, you can go back to uh, Bruno, the story of Bruno, who accepted the Copernicus idea about the centrality of the sun and the solar system, instead of the Earth that the church preferred. He was burned at the stake. They don't do that now. They
Yeah.
don't burn at the stake anymore, at least as far as I've heard.
Yeah.
But there are other.
It's a social exclusion zone. It's a
Yeah,
social exclusion zone.
yeah, social exclusion zone. And there have been a half dozen papers that I know of that have challenged what we've done, and I I responded to the first of them, I think, adequately, but some of the people are not interested in really exploring the evidence and exploring both sides, which you know very well.
Yeah.
Uh, you know they 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 have a tendency to want to stick to the status quo, and often the status quo is just simply not right, and is justified. On the other hand, the flip side is that interest in the fourth phase water has just exploded. There
Yeah.
are now so many people who are interested from so many different disciplines that I'm basically traveling more than I'm home because so many people are interested in hearing about this stuff, and it has. I was just at a conference in Sweden, and after I gave my presentation, there was a second presentation, and the question was, if you have $1 billion, what would you do? And of course, I'd be delighted and thrilled to have $1 billion, but it's really difficult to get the money for work like this that runs against the mainstream.
Mm.
But I was able to articulate in 15 minutes the multitude of different areas that are impacted by this finding, assuming it's right. It goes from health to all kinds of technologies that
Yeah.
come out of this that we have no ability to pursue, because in order to pursue it, you need people, and in order to hire people, you need money, and money
Mhm.
has been hard to come by.
Mhm.
We can't depend on the government because it's almost impossible. The
Yeah.
National Institutes of Health, I think the word water is like a foreign language for a foreign word for them.
Mhm.
It's not in their lexicon.
Mm.
Uh,
But
they don't know that word. So it's hard. So it depends on private donations. And we've had in the past, we've had one magnificent donor, a billionaire, who said he would supply funding uh, indefinitely, very major funding, unless he ran into a financial uh, um, problem.
mm.
After 4.5 years, he ran into a financial problem. He gave me 1 year's notice. And he still signs his emails, love Peter. He's really enthusiastic about everything we do, and he's
Yeah.
helping us to get other private money, but it's
Yeah.
been a real challenge.
Okay. So
So
if someone is listening to it, you have ideas how we can make support to your institute. Yeah.
just send me an email.
Yeah.
My email address is all over the internet.
Yeah.
I'm very reachable, and
Oh, great. Thank you. And you are responding. I also could find out. Gerald, to come back to this special detoxification possibility, because when we are saying that this easy water is excluding toxic things, what about if we put easy water into a cell where something toxic, like spike proteins, for example, could be? Could it be that the easy water would exclude this topic out of the cell?
Well, we haven't studied that, so I can't say. In theory, or in principle, it could be, but
Mhm.
if, for example, the spike protein sticks to another protein, it may be difficult
Mhm.
to get it out of the cell. I don't know enough about that topic to
Yeah.
comment sensibly on it.
Mhm.
I wish I did, but
Yeah. Were there in the exclusion zone is when there's a virus, for example. Yeah, if there's a virus, DNA, mRNA, mRNA, a messenger RNA, is it excluding alien DNA or?
we don't know. We've never
Okay.
studied it.
Okay.
I'm really sorry to be unable to answer those questions.
No, no.
Just that, in Seattle, the day has 24 hours, and, and there are so many things that we're doing, and that's not been one of them.
Yeah.
It could be, and maybe somebody else could study it, but we haven't done so.
No, no. It's working on hypothesis because it's a very big problem. Here in Germany, you have about 8 million people who have the problem with post-vac or post-vaccine, and I think in America it will be also a huge amount of people that have the problem, and they even don't know. So, but they have yeah, they have injuries, and also cancer is discussed about that topic. And the questioning is because all scientists have the problem how to get rid of the spike proteins from the virus, but also of the vaccine, because it was the same programming. Yeah.
Yeah.
So, and when these mRNAs and DNAs are coming into the cell to produce always these spike proteins, so they transform and then produce it. So the question is if there's an how to get rid out of this cell from this special messenger RNA that becomes a DNA.
I wish I could help.
Yes,
I
okay.
can't. Oh.
So the detoxification, that what did you find out of detoxification by the easy water in the cell?
Well, we haven't studied detoxification. I I wish I could answer those questions. They're highly relevant. It's just
Mhm.
that, you know, we 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 can't do everything, and somebody should address that question. I think it's really
Ah,
important because there is a possibility that the easy water could be responsible
mhm,
for detoxification.
mhm,
Because it does undergo, if we're right, and I believe we're right, it does undergo a phase transition. And during this phase transition, it goes from easy water, fourth phase water, to ordinary liquid water. But then easy builds again, and as it builds again, it should be able to push out any toxins or other molecules that are in the cell that are extraneous. But how this happens exactly, and if it happens, and it all remains to be explored.
So it's called exclusion zone water because it is excluding what did you see? What is it excluding?
Well, when we first did experiments, um, we were looking for structure in the water, just like Gilbert Ling,
Mhm,
and if there's uniform structure of molecules, if it's a liquid, pure liquid crystal, it as it forms, it should push out solutes. And so we started with little spheres called microspheres, mainly because you could see them in the microscope. And so we could visualize these microspheres getting excluded from a particular zone of water. That zone was next to some hydrophilic surfaces.
mhm,
But after that, we tried many different substances, other particles, all kinds of molecules, various dyes, and they all got excluded. Uh,
mhm,
we we can't easily monitor very small substances, but as a class, we could determine that virtually everything that we studied got excluded. Um,
mhm.
so that's why we called it exclusion zone. It was the advice of a colleague that, instead of going through elaborate descriptions, why don't you just give it a name? Call it exclusion zone because it excludes. And it worked beautifully in the US because exclusion zone EZ. But in
Mhm.
Germany, it's EZ. So EZ is easy to remember, but EZ is not so EZ
Right.
to remember, if you know
Yeah,
what I
but easy going is already a good frame for Germans
mean.
too. So
Okay, easy going. Right. So you understand.
yeah. Yeah, that's fantastic that what you're telling us about this special phase of water and also about biophysics. So thank you for that. If there's, as you already said, it's this idea is transported to many people and worldwide they are working on this topic and they also are already producing products about that. Is there any knowledge from your side that when we take easy water, when we drink it, or how can we do something good for our health?
There are at least a half dozen simple expedients, and maybe we could terminate with that because I need to
Yeah.
go. Yeah. So uh, you need easy in your body. You need easy in your cells. For a cell to become or to be healthy, it needs to be filled with easy water.
Mhm.
We have various evidence for that. And the way you get easy water yourself, one way is to drink water. And some of the water gets translated into easy water. Some gets peed out, but keeping well hydrated is one way to do it. Another way is to do so-called juicing. I don't know what it's called in Germany, but you go to the backyard, you take some freshly grown leaves of plants, and you squeeze the juice out of them, the water
Mhm.
out of them. And there are machines that you can purchase to do that. And what is that juice? The juice is the water from inside the plant cells, and the plant cells are freshly grown. They're filled with easy water. So you're basically drinking easy water. And that's one second way to replenish what's missing in your cell, in your cells. Third way um, is to expose yourself to sunshine. Now, in Seattle, in the wintertime, we don't see the sun very much. But when the sun pops out, people have smiles on their faces. And, you might surmise that this is a psychological effect. And indeed, it probably is to some extent. However, the sun, half of the sun's energy is infrared. That's why it feels warm. And infrared can penetrate your skull. If it does penetrate your skull, and there's clear evidence that it can, because you can do imaging of the brain from an outside source of infrared. So it's got to penetrate. So if it penetrates, then it builds easy water in your brain cells. And yet then your brain cells are working better. And if you're depressed, you get undepressed, and et cetera, et cetera. So exposing yourself to sunlight is a positive action that you can do. The extreme of that light is the sauna, or as the Finns say, sauna, because it's basically heat, whether it's dry or moist, it's heat. And heat is sort of equivalent to infrared energy. So you're getting infrared energy in that way. Let's see, there are
Yeah, that's
I mean those are a few. Oh yeah, there are certain agents that have been known since Ayurvedic times to be good for health. That's like 5,000-10,000 years ago. We're talking turmeric, basil, ghee, clarified butter, and we tested all of those, and we found that of the 7 or 8 that we tested, every one of them builds easy water.
Oh, yeah, interesting. Yeah.
Yeah. So those are, I'll just state one more, and that is hyperbaric oxygen therapy, which is not so easily available, but you get into a tube and you're exposed to high energy and high oxygen. And we found experimentally that both high oxygen and high pressure both build easy water. So the reason why hyperbaric oxygen is so powerful is that it builds easy water. So those are some of the expedients.
Very interesting. All solutions you have said now are helping with with post-COVID and post-vac. So it's interesting that
All right, I didn't know that.
exactly the same topics you summarized now. It's really interesting.
Wow. Well, that's so interesting. Thank you. Yeah.
Yes. So thank you very much. I'm very happy to have you here, and I hope we stay in contact and maybe
Sure.
have a
Absolutely.
second.
Yeah. Okay.
Was a
Thank you.
wonderful.
Thank you, Derek. Okay.
Thank you very much, Gerald.
Okay. Okay. Bye-bye.
Bye.